So really, it's a pleasure for Euro-DyMA to welcome all the attendees today for this first Euro-DyMA Pharma's Day. As I mentioned earlier, this program will be broadcast tomorrow at the Awareness Day of Myotonic Dystrophies. And before the core of the subject, I will give you an overview of Euro-DyMA at the current time. So why Euro-DyMA? Well, you know, in Europe, there are many people affected by DM1 or DM2. How many? I can say between 50,000 and 100,000, probably. Most of the countries have nationwide patient associations, large, small, depending on the country. There also exist some European non-specific teams dealing with DM1 and DM2. There are these ERN from the clinical side, TreatMD, ENMC. And also, you know, and you are well aware of that, that clinical trials are knocking at the door for very potentially decisive molecules. And also, you know, the very specific cognitive issues in DM. So it's more or less critical to associate deeply patient groups to ongoing trials. Also, the experience from other federations showed a real gain in time for the drug design, the trial and approval when there is a federation inside, for example, Europe or the US. So Euro-DyMA is the only non-profit European patient association fully dedicated to myotonic dystrophy type 1 and type 2. Our roadmap. Well, at first, bring some expertise or some experience of DM patients and family to European scientists, professionals, drug developers, and health regulators. Improved, harmonized knowledge about the disease in Europe, and there is a lot of job for doing that because it's really unequal from country to another one. Then identify unmet needs in Europe and promote care and research programs. And we are now doing that carefully in detail in the next weeks. Stimulate, aggregate collaboration between patient organizations. And additionally, we would like to help creating a network of European young clinicians highly committed. And we have a golden rule, which is to make sure that we keep efficiency and flexibility. We are a small team. We are very committed in myotonic dystrophy, and this is really positive for our disease. So our members, we have France, we have Germany, Belgium, Italy, Denmark, UK, Cyprus, Greece, Netherlands, and Switzerland. Here we have the family picture, which was taken end of 2019. Some people left, but we will do another one as soon as possible with the current board of Euro-DyMA. We are 18 at the time. We have a scientific advisory board with Basil van Engelen chairing the SAB, Dennis Furling from the Institute of Myology, Darren Monckton from Glasgow University, Guillaume Basset from Institute of Myology, Benedict Schrözer from Germany, and Eric Ekström from Sweden, Giovanni Meola from Italy, and George Papadimas from Greece. We also develop a library because, you know, there are many documents dealing with myotonic dystrophy, but most of these documents are not really aggregated except in MyoBase. But MyoBase, which is a database which has been developed by AFM in France, but it's dedicated to all the diseases. And so we have asked AFM to develop a subset of MyoBase, which is accessible from our website. And it gives you access to more than 4,000 documents and links in direct access in various languages. You have a picture, a snapshot of the screen, which that you can see on the website. There is also a searching engine. The objective of that is to make sure that if you need info, you get it immediately and for most of the document at no cost. Okay, so the news about Euro-DyMA. Well, it's now well installed in the DM community. We are members of Euro-DyMA. We participate, we have a member in the clinical network PRN. We participated in ENMC workshops, and we have established strong links with Myotonic Dystrophy Foundation in the US. And there is one point missing, which is drug industry, and it's the reason for this meeting today. We will start to discuss with you. We are involved in at least 3 programs which are in progress in Europe. And we also participate or organize events dedicated to DM1 and DM2. At first, the Global Alliance for the International DM Awareness Day of tomorrow. We had a scientific workshop in June, which was very efficient. We have obtained a snapshot of the care situation in Europe, starting from the patient perspective, which is a bit new. And we have reviewed and prioritized unmet needs in care in Europe. And we are starting to correct this action and to update it. Of course, the Pharma Day today. We also tried to set up an awards of the best scientific paper of the community. It was unsuccessful this year because it was announced too late, but we will come back next year and we will grant a paper and with some money joined to that for the best scientific paper. And of course, we will participate in IDMC if IDMC is confirmed next year in Japan. So the Pharma Day, why? Well, at first, create with the drug industry the same link as we did with academic teams and patient organizations on the base of a win-win process, which means we bring efficiency and flexibility. And of course, we get also information which is updated from your side. We want to share simultaneously in Europe update and info about the drug development and trials, and also identify the industry requirements in terms of patient participation to organize our association. You know, we are only 18. We are very well, we are busy in our respective associations, which means that we have to tune carefully what is needed from the industry, from us. And that's also a question that I will ask to you at the end of each talk. What is your requirement in terms of patient participation in your program? And the meeting agenda. The meeting agenda, first paper is from Roberto Massa from Italy about metformin. The next one is from Beatrice Lemouzy from Artex about miR-23B inhibition. Mike Snap for AMO with the tidiglucib trial. Caroline Gaufret from PepGen. Unfortunately, they are not with us today, and they have recorded their talk. It takes only 10 minutes, so it means that we have a lot more time than expected at the beginning. So, and then we will finish with the DyMA talk from Ashish Dugar from DyMA. We should take the time. It was expected to be 30 minutes, questions included. We can go up to 40 if necessary. If it's not, we will have time to end the meeting earlier. And that's all what I had to say. Arrêtez la présentation. We are on time. And the next speaker is Beatrice. Just pas que je suis conversation Microsoft. Artex partagé. And that's it. Do you see the slides?
Yeah, sorry, I was on mute.
Okay.
Yes.
Are you successful in taking the control of the meeting? You have something on top of your screen to move the control to you.
Let's see.
Sometimes it works, sometimes it doesn't.
No, sorry, I don't. Well,
You don't see
yes,
it?
yes, yes, yes. It
Okay.
seems it's yes. No, it's not working very well because now I
like
don't see anything.
Everyone's the slide. Okay.
I don't even see the slides right now.
Oh. Don't see the slide?
No, I don't know if it's my problem because previously when you were also presenting, sometimes I lost the slides. I don't know.
Okay, so what you could do is open your file, and I will give the number of the slide.
Okay.
Sorry about that. So slide number 2.
Yes. Okay, well, I will present myself. Well, hello, good morning, and thank you for inviting me here. It's a pleasure to share our project with you all. Well, we are a small company of the University of Valencia, a spin-off. I was previously a researcher at the university. I was working with myotonic dystrophy for about 12 years until we have some results that we believe that they deserve to move forward. So then we created the spin-off to do it. This is our team. As you can see, I have some people helping me at the management level. We are located in the Parque Científico of Valencia, and we also have a laboratory in the university. We are still building our team. The company was just created at the end of 2019, just before the COVID. So we are still growing. I'm not sure if you can see that we have a board of directors with includes 2 venture capital, In Vivo Capital and Advent France. And also in our scientific advisory board, we have experts in different fields, such as oligonucleotide therapeutics, such as Ramon Erija and Eric Markusson, that have been very helpful in the design of our antisense molecules. And we have also 2 clinicians working on the field, very well known in myotonic dystrophies, such as Nicholas Johnson in the University of Virginia and Guillaume Bassef here today, that is located in the Institute de Myologie in Paris. If you move to the next slide.
So next slide is number 3.
Number 3.
The problem.
Yes. Well, probably you all know better than myself what is myotonic dystrophy. It's a rare genetic multisystemic disease. Just to let you know that although the genetic cause is very, very well known, there is no cure against this disease. That's why we are here today. And well, just to focus on myotonic dystrophy type 1, as I am the first one today, I will have the pleasure to introduce the mechanism. It's a very complex disease, but to make it short, I will tell you that it's due to the expansion of a microsatellite in a non-coding region of the genome, which means that this CTG triplet is expanded and in the patients, creating an RNA which is longer than usual. And this extra long RNA, it's folding into a hairpin that is retaining MBNL proteins. So most of the symptoms are directly caused by the depletion of MBNL proteins, because these MBNL are important regulators of splicing and RNA metabolism in general. So once they get sequestered here, they lost their activity. And several approaches have shown that the overexpression of MBNL could be therapeutic. So that's why we focused on this protein. And if you go to slide 4, you can see here that, sorry? Sorry? Yeah, in slide 4.
Slide 4, okay, yeah.
Yes. Yeah, you can see the mechanism of our RNA therapy. As I told you, we were focused on MBNL. And what we discovered in the laboratory is that MBNL levels in the patients are depleted by two different mechanisms. The classical one that I told you is the retention into the RNA, extra long RNA or the mutated RNA. But we have also observed that there are some inhibitors of these proteins that are upregulated in the disease condition. So in the muscles of the patients and also in different models of the disease, we have observed that there are microRNAs that are upregulated. Here, MIR23B and MIR218 are upregulated in the patients, and they are directly blocking the production of the protein. So in the patients, these proteins are depleted by these two different mechanisms. And what we have observed is that just blocking one of them, which in our case is the microRNAs, we obtain enough levels of protein to have a relief of the symptoms. So as you know, most of the companies working on the field are trying to destroy the toxic RNA or inhibiting the interaction between the protein and the RNA. But we are acting on the other part of the problem. We are blocking these microRNAs using antimers, which are single-stranded RNA that bind to the microRNAs and block them. And we obtain proof of concept of this therapeutic approach in different models of the disease, such as patient cells, and also a mouse model, where a subcutaneous injection of these antimers produce a long-acting therapeutic potential. So we have already 2 papers published and one about to be published with this proof of concept. And if you go to slide 5, I will show you what we have been doing since we created the company. Well, this slide has several levels, so you can click until you see it complete. We have been focused on selecting one lead molecule, which is the most effective. And so we started with an in vitro screening of molecules, all of them anti-microRNAs that can inhibit either MIR23B or MIR218. And we started with screening more than 100 different antimers, tested them in the human DM1 cells. And then with the best molecules, with 23 hits, we started testing them in the in vivo model, specifically in the HSA-LR mice from the laboratory of Charles Thornton. In the first preselection, we tested them at a very high concentration, just trying to get rid of those that might be toxic. And then a group of them were tested at a very low concentration, just looking for the most efficacious molecules. In the HSA-LR mice, but also in healthy mice to see the activity in control mice. From all these studies, we have selected a lead molecule against MIR23B, which is ATX01, which was the most efficacious molecule. And we also have a backup against MIR218, just in case we observe any toxicity related to the sequence of the MIR23B. In the bar graph, you can see a comparison between the effect of these lead molecules and the previous antimers when they are administered to HSA-LR mice at the same concentration. So you can see that the lead molecules in pink have a stronger effect in strength. In fact, they are normalizing the strength of the mice, and also stronger effect in myotonia than the previous molecules. So our molecules, which in this case, the lead molecule is conjugated to a fatty acid, is increasing importantly the efficacy in these in vivo models. If you go to slide 6, I can show you the type of experiments that we are doing in the mice. As I mentioned, we are using the HSA-LR mice. You can see here in this publication from Charles Thornton a description of the phenotype of these mice. The mice are expressing more than 200 CTG repeats. So this will be more a model of the adult onset of the disease. They show muscle histopathology, grip strength reduction, missplicing in the muscles, and also myotonia. And you can see here an example of one time response experiment in the DM1 mice. Here we are, I am showing you the results of our lowest effective dose in the mice, which is 3 mg per k. One intravenous injection of these anti-mirs at 3 mg per k, and then the effect were evaluated after 5, 10, or 15 days. So we can see the duration of a single injection. You can see in the functional rescue experiments, PNF, here it's the percentage of normal force. So comparing the mice which were injected with PBS as a control, which is the gray line, and the mice injected with our product in red, you can see that there is an important improvement in the strength during at least 10 days, and then they decrease after 15 days. Similarly, with the myotonia, there was a decrease in the myotonia during 5-10 days, and then they turn back to the normality. And at the molecular level, we are evaluating in the gastrocnemius and quadriceps muscles. Here, gastrocnemius is GT and quadriceps QD. Evaluate the levels of MBNL proteins, and also down you can see some splicings which are regulated by these proteins, such as the ATP and also the chloride channel. So as you can see, with the protein, there is a strong upregulation of the protein after 5 days, and then the protein decreases after 15 days, similarly with the splicing. So we know that at our lowest effective dose, we have at least 10 days of efficacious reply in the mice. And with higher doses, we achieve more duration of the treatment. If you go to the slide 7, I have summarized the advantages of our therapeutic approach. As you can see, our main differential factor is that we are targeting a different molecule. Our new target is the microRNA, in this case the miR23B. These microRNAs are located in the cytoplasm, so they are easier to access using anti-mirs than the DMPK, because in this case the DMPK foci are inside the ribonuclear, are inside the nuclei. And in our case, well, this strategy could be compatible with others. We are going in the same direction, so they might be synergistic. Please go to the next slide, and I will give you some details about the relevance of our target. Please go to slide 9. I will tell you something more about miR23B, because the advantage of using this microRNA is that it is very well conserved among different species. So going from the mice, rats, mini pigs, dogs, and also humans, of course, it is completely conserved. The sequence is completely conserved. So we know for sure that the molecules that we are using in the proof of concept studies could be the same molecules that we apply to the humans. In addition, can you see the graphs already? You see the complete
yes,
slide?
you are displaying the full slide.
Okay, because here you can see that not only the microRNA is conserved, but also the mechanism of action. If you see in the human cells, when we use ATX01, there is an almost complete depletion of the levels of miR23B. And when we decrease the levels of 23B down, you can see that the MBNL1 protein increases. And similarly, in the rat cells, when we use anti-miR against 23B, the depletion of the microRNA increases the protein, and the same with the mini pig cells. So these two species that we are doing in the preclinical studies, we know for sure that the mechanism is conserved. So the whatever toxicity or efficacy we observed in this species could be easily translated into the studies in the patients. Please go to slide 10. And we believe that this microRNA is a relevant target for several reasons, but one very special is that it is consistently upregulated in different DM1 models, such as you can see here the mice muscles, gastrocnemius and quadriceps. This is a mice which is a HSALR, a model of the disease. Also in human muscle biopsies, and also in cells, here the PDM. In cells expressing the CTG repeats, the microRNA is also upregulated. And it is even more upregulated in cells which are expressing longer repeats. If you see the graph on the left, what we have done here is to overexpress CUG repeats of different lengths in HeLa cells. And here we observe that just overexpressing the naked CUGs, we obtain an upregulation of the microRNA, which is also dependent on the length of the repeats. So the higher the number of the repeats, the higher upregulation of the microRNA. And that means that there is a strong correlation between the mutation itself and the microRNA levels. And we are studying in the laboratory the consequences and the causes of this relationship. Please go to next slide. I will talk to you here about another advantage of our therapeutic approach, which is that this is a multi-target approach. In slide 12, you can see that we have discovered that our anti-microRNAs, in addition to upregulate MBNL, they are decreasing the levels of the DMPK. This was an observation first in human cells, in human DM1 cells, in RNA-seq experiments. You can see that our previous antimers, here DM123B and DM1-218, when we treat the cells with these antimers, we are reducing the levels of DMPK expression in comparison with a DM1 cell treated with a scramble antimer as a control, which will be here the gray box. We have validated the results with a different technique in qPCR and with different lines, with different cell lines, and obtained the same results. And we have also performed these experiments using our lead molecules, which is ATX01, and the backup molecules, which is ATX02. As you can see, we obtain a reduction of almost 85% in the human cells. So we are also targeting this very important player in the disease. Go please to the next slide. And here I wanted to tell you more about our molecule, because as I mentioned previously, our lead molecule is not only an oligonucleotide, it is conjugated with a fatty acid. And we are doing this conjugation because it's improving the efficacy of the oligos in the muscles. If you see in slide 14, here we have compared our lead molecule, which is ATX01 on the left, and the naked version of this oligo, which is only the oligonucleotide without the fatty acid on the right. When we treated HSALR mice models with intravenous injection of these molecules at 3 mg/kg, and then observed the MBNL levels in different tissues, we see that with our lead molecule, with ATX01, we have a significant strong upregulation of the molecule of the protein in quadriceps and gastrocnemius, and also in the diaphragm, but not in other tissues such as the kidney or the liver. This is important because oligonucleotides tend to accumulate in the kidney and the liver. So these might be the organs that might be more strongly affected by therapies with oligonucleotides. And in fact, some adverse effects are specifically due to this accumulation. If you compare this data with the data of the naked oligo, these oligos have a mild effect in quadriceps, but not in gastrocnemius, not in diaphragm, but have a strong effect in the kidney as a result of the accumulation. So what we are doing with this conjugation of the fatty acid is to driving the oligo to the muscles and avoid potential adverse effects in other tissues. Please go to slide 15. We have already done some toxicity preliminary studies with these oligos, some in vitro studies which are which are especially helpful in therapies with oligonucleotides, such as testing effects on coagulation, on complement activation, potential activation of cytokines, and also a panel of pharmacological off targets or their effects in potential hemolytic in human plasma. With all these studies, we observe that our molecules are safe. And we are also starting some studies, toxicity studies in vivo in different models. We started already the experiments with the rats. But until now, I put here this traffic light in green because all our studies until now is telling us that we could move forward with them. So go please to slide 16. I will show you here some results we obtained in the rats. These are control rats, but the good thing about our therapy is that we can follow the levels of target engagement of our therapy because we can measure MBNL1 upregulation even in the control animals. So here in the rats, we can measure which is the effective doses just by measuring MBNL levels in the gastrocnemius of the rats. Here we have done a first experiment, which is a dose range study in the rats, injecting intravenously at different concentrations, 3, 6, 8, and 30 mg per kg, injecting once per week during 3 weeks. And as you can see in the graph of the dose response, here we are measuring the levels of MBNL1 protein. And as you can see, the first efficacious or the minimum efficacious dose was 8 mg per kg. And here in these efficacious doses at 8 and 30, we have not observed any adverse effects. In the following study, we have compared intravenous and subcutaneous administration at the same concentration and 8 mg per kg. So here you can see in the graph that with intravenous, IV or sub-Q, we obtain similar levels of protein in the muscles. So that means that subcutaneous could be a potential route of administration. And in the following graph, we have done here a study of the duration of the effect after one single dose. So after a single administration of 8 mg per kg, we can see that there is a peak of maximum effect after 5 days, but still after 22 days, we still have at least twofold of the protein. So at least 3 weeks of duration of the efficacy with this dose. We continue now with more experiments in the rats, and our idea here, you can see the development plan in the slide 17. You can see our development plan for the future with this product. Our idea is that according to all our plans, we will be completing the preclinical package needed for phase 1/2A by the end of 2022. So our idea is to submit the IND or CTA at the beginning of 2023. So when we will be able to initiate the studies, the first in human studies. We have already had a first interaction with the regulatory agencies with a pre-IND meeting to validate our regulatory roadmap. We are about to apply the ODD in US and Europe. And as I mentioned, our idea will be to initiate the clinical trial at the beginning of 2023. This will be phase 1/2A. and then to do a potential phase 2-3 in 2024, which might be registration. And well, thank you very much. This is all the information I have. Hopefully, in a year by now, I will be telling you good news and telling you that we are about to submit the clinical trial application. Well, I'm open to your questions, and thank you very much for your attention.
Thank you so much. Very exciting paper.
Thanks.
You have any questions? No question at all. Wow.
Everything very clear.
Yes, okay, yeah, one. Do you need some patient organization participation to help to speed up your studies, to help obtain commitment of patients or something like this? Do you need some help, some assistance from us?
Yeah, well, right now, of course, the communication with the patients is crucial, especially after the first interaction with the regulatory agencies we had. It seems that for them it's crucial to see in the clinical studies that the endpoints correlate with a clinically meaningful functional improvement for the patients. So this is something very important to know, that it is not enough to measure an improvement in one parameter, which makes no sense. And in this sense, we will work very hard for this. We are still, unfortunately, far away from the clinics, but our chief medical officer is joining by the end of this year. And she will work very hard on this, because we really need to assess, or we still need to define which endpoints we will need. And in this, we believe that here we will strongly need the participations of the patients to see what is really, what means clinically meaningful for the patients.
So it means you may need some contact with us mid-22?
Yes. Mm-hmm. At the beginning of 2022, when we start defining the clinical trial and deciding the endpoints, that might be very relevant, yeah.
The clinical trial will be limited to Spain, to Europe, for all the world?
Well, we have an agreement with the clinical network, myotonic dystrophy clinical network, that has sites in US and Europe. So our idea is to start in the US and then move to Europe in the phase 2.
Okay, very good. No other question?
Alain, I have a question.
Yeah.
Hi, this is Denis Furling speaking. How are you?
Hello.
Nice talk.
Yes.
Just a question about the MIR23. What are the other functions of this MIR?
Well, it depends on the tissue, but there are other. It has been described that it, for example, it participates in the differentiation. It has a more important effect on differentiation of several tissues. So that's why we believe that using it in adults will not have a strong impact on the effect of the microRNA, because it, as I mentioned, it's more involved in the differentiation, for example, of the white blood cells, and also in the kidney tubular cells. Mm-hmm.
Okay. And so just a second question. You see a massive decrease of the MPK expression. Do you have any explanation for that right now, or just an observation?
Yes, no. Now we are working in the laboratory of Valencia. They have a project focused on this, because what we have seen is this relationship with the repeats and the expression of the microRNA. And we have also observed that the downregulation of the protein of MBNL is also increasing the microRNA. But we still don't know the connection. So we are testing different hypotheses because the DMPK is not a direct target of the microRNAs, which might be the first thing to test. So we know that it's not a target of the micro, but we still don't know which is the connection. We have observed in some congresses, for example, I think that the tidoglucib is also decreasing DMPK. So we believe that there is a kind of internal loop with some of the players in the pathogenesis, but we still don't know the mechanism. That will be something very interesting to check.
Okay, thank you.
Thanks.
No other question? Okay, thank you so much. I'm waiting for the next year, which will be very exciting, I think. Okay, let's jump to second talk, which was in fact from the University of Roma. So we have been obliged to move to leave OneDrive from Teams. And do you see the slide right now about metformin?
I see the slide. It's okay.
Okay, good. I'm sure that you cannot run the slide because it's on my computer. So I will do that like for the previous one. Okay, let's go.
Yeah. Okay. Let's go back to the first one. Okay. Good morning, everybody, and thank you, Alain. Thank you, Giovanni, for your kind invitation to this exciting Pharma's Day, which is something really new for the myotonic dystrophies, at least. Exciting because until a few years ago, it would have been almost unreal or utopistic to talk about in practice, I mean, in practical terms, to talk about treatments for the myotonic dystrophies. Of course, we have experienced several attempts to address this problem in the past years, in the past decades. And of course, what we've done is to start with approaching this problem with the possibility of treating single aspects of these diseases, which are very complex, of treating single symptoms. So we have had a few, I would say a few successes in doing so. And I mean, I refer to the use of mexiletine, for example, for myotonia. Which is a useful drug not only in the congenital myotonia but also in the myotonic dystrophy to some extent. And I refer to the use of modafinil and central nervous system activators to treat sleepiness, daytime sleepiness, and this is quite successful. However, as I am going to remind everybody, the myotonic dystrophies are a very complex and widespread diseases. So next slide. Manifestations, clinical manifestations, as we all know, are widespread in the body. We have cognitive disturbances. We have sleep-wake cycle alterations, which are very important for the quality of life of the patient. We have typical facial features with baldness, ptosis, temporal muscle atrophy, and more extensively a general muscle atrophy. We have eye disorder with cataract, and then we have heart and respiratory problems, which are the main causes of death, finally, of these patients, with cardiac conduction defects, arrhythmias, which are very dangerous for the patients, and we have as well respiratory problems, which become very problematic, especially during general anesthesia and during infectious diseases of the respiratory system. We have dysphagia and a wide range of disturbances of the gastrointestinal tract, with dyspepsia, with slowness of the emptying of the bowel, and with irregular emptying of the bowel. And we have gallbladder stones and many other manifestations. And then we have endocrine problems with insulin resistance, less commonly diabetes, but we have often hypothyroidism, hypogonadism, and generalized low assets of vitamin D, and sometimes a metabolic syndrome. Finally, recent research focused on an increased oncological risk, especially with some type of malignancies, besides other tumors like skin tumors. So DM1 and to some extent DM2 are really widespread diseases which involve the whole body. The next one, please. Here we see, just to remind everybody, the core features of DM1, which are a typical phase with apoptosis, with weakness of facial muscles, and with atrophy of temporal and masseter muscles, and the typical baldness. We have the iridescent cataract in the posterior pole of the eye of the lens. And we have the typical distribution of distal atrophy and weakness in the four limbs, and in the neck and face muscles. Of course, this is relevant to DM1. When talking about DM2, we have to move from distal to more proximal muscles, and facial muscles are less severely involved. And then we have, of course, myotonia, and which can be, triggered by percussion of muscles or just by action. So it's a grip release myotonia, which is very evident in the hands. The next one. However, DM1 and DM2, although similar, are not the same diseases. They share a common pathogenic mechanism, but this mechanism somehow targets in a different way, or to a different extent, some tissues and organs. And for example, we have a different severity of weakness and atrophy in different groups of muscles between the two diseases. We have less common cardiac defects in DM2 versus DM1, and we have less common sleepiness and apathy in DM2 versus DM1. Whereas, for example, insulin resistance, hypothyroidism, in general endocrine problems, and cataracts are almost present to the same extent in the two diseases. Moreover, and more important, a congenital form which is known to be present in DM1 is not present in DM2. This makes DM2 to some extent a less severe, at least in most cases, a less severe disease than DM2, and a disease that usually has a later onset as opposed to DM1. Okay, next one, please. Okay, this is just to show how we can gather information about DM1 and DM2 patients. We have a Roman, so-called Roman database, which is made by neuromuscular centers active in the Roman region, in Lazio, in the Roman region, which is a region of about 6 million inhabitants. and with 4 main neuromuscular centers. And this is our common database, which gather, as you see, information about clinical and genetic information about more than 800 patients, with a ratio between DM1 and DM2 of about 13. This is quite different from the ratio which is reported in Central and Northern Europe, where the prevalence of DM2 seems to be much higher than in the Mediterranean countries. As you see, there is a slight prevalence of male patients in DM1, and a slight and the opposite in DM2. The mean age at onset, as I mentioned before, is really different. It's in the early 3rd decades for DM1 and early in the 5th decades in DM2. And symptom at onset is most commonly myotonia for DM1, but more commonly refers to other neuromuscular symptoms in DM2, for example, muscle stiffness and myalgia, which makes diagnosing DM2 more difficult at the beginning. Next one, please. Now, pathogenesis of the myotonic dystrophy is common, although some differences are recognized. And the mainstream mechanism calls for a toxicity of the mutant RNA in both diseases. RNA is produced by the expansion of the unstable triplets in DM1 of the quadruplets in DM2, leading to an accumulation of the RNA within the cell nuclei, the so-called foci, so this mutant RNA does not exit the nucleus, and it stays, and it makes a sequestration of the CUG binding proteins like MBNL, and therefore leads to an altered splicing of numerous mRNA, which are regulated by the CUG binding protein. And this mRNA has so far been described as being prevalent in muscle, heart, and brain tissues, but for sure they are important also in other tissues and organs. Next one, please. So we can call this thing a spliceopathy. It's not a very nice term, but you know, now it's been accepted by the scientific community. So toxic RNA produces a spliceopathy through the accumulation of RNA within the nuclei. And here we can see, I mean, it's a little dark, but we can see little red spots indicating toxic RNA, which is made visible by, of course, by a fluorescent staining within the nuclei of muscle cells. So this spliceopathy, this is a list of RNA and the respective proteins which are misspliced in the DM. This list is not updated, so there are many more now. But most of these proteins are important for skeletal muscle, and some are important for the heart and some for brain. But there are, as I say, there are many more now. Next one, please. Among the numerous recognized splicing defects, one affects the insulin receptor. This defect is characterized by the expression of a fetal isoform, which is called insulin receptor isoform A, which has a lower affinity for insulin, so it has a low functional power. So this prevalent expression of the fetal isoform versus the adult isoforms causes an insulin resistance, sometimes inducing insulin hyperproduction, and downstreams contributes to muscle atrophy. We see here in a nice experiment by the group of Giovanni Meola how the insulin receptor A is hyper-expressed in DM1 patients versus the common prevalent expression of the IRB in normal controls and other myopathies or muscle disorders. Next one, please. However, these splicing defects affect multiple functions in the body, in different tissues. So beyond the insulin receptor, we know that there is a missplicing in the expression of the muscle chloride channel, which regulates influx of chlorine within the muscle fibers, regulating the excitability of muscle membrane, and this leads to, at least as a main cause, leads to myotonia. But we also have missplicing of the cardiac protein troponin T, which probably contributes to the cardiac abnormalities, cardiac conduction, and even to cardiomyopathy. And there are missplicing of other intracellular muscle proteins such as RIR1 and TMR1, probably contributing to the muscle weakness and wasting. And there is missplicing of important neuronal proteins in the brain, such as the tau protein and the NMDA receptor 1, which probably contribute, but this is a very complex mechanism, which is mainly unknown so far, contributes to CNS effects in both DM1 and DM2. Next one, please. Now, of course, one could look for possible countermeasures for these functional defects produced by splicing derangement. The widespread involvement among tissue and organs in DM1 and 2 limits the efficacy of treatments that was downstream the pathogenic mechanism. So only treatments that may address the problem of splicopathy as a whole, so upstream, may be effective, that is curative. Next, please. Recent evidence shows that the drug metformin, which is used for decades now to treat diabetes as a first-line treatment for diabetes, is able to correct several splicing defects in human cells with the typical DM1 triplet expansion. And this effect is obtained at common therapeutic dosages of the drug, which is a encouraging new, of course. So metformin has therapeutic potential for diseases linked to splicopathy, as we can see here in this very nice experiment by the French group at Institut de Biologie and colleagues. The expression of some splicing products we see here on the right column is brought back to a similar to normal extent, as opposed to the increased expression of the aberrant isoform in the untreated cell. This is true for the troponin T here. This is true for the chloride channel here, and this is true for the insulin receptor here. So the next. Okay, this is more or less is recapitulating the previous one, so we can go on. And this is not all, because recent evidence from the Spanish group of Adolfo López de Muñain shows that metformin can also reverse some metabolic and mitochondrial defects and some aspects of an accelerated aging of cell, which has been shown to occur in especially in DM1 patients. The DM1 patients has several aspects of the progeria, so of an accelerating aging of cells and tissues widespread in the body. And metformin can reverse some of these aspects. So this is maybe an additional therapeutic target besides spliceopathy that can be derived from the use of metformin in DM1. Next one, please. So moving to human studies, somebody already has done this, and the group of Guillaume Basset in Paris has performed a first trial in humans using metformin in patients with dystrophy, with myotonic dystrophy type 1. This was a phase 2 study, double-blind, placebo-controlled, on about 40 DM1 patients from a single French center. They administer 1 gram metformin or placebo 3 times a day for 40 weeks, which is almost 1 year. And the primary clinical outcome was a change in the distance walked at the 6-minute walk test, which is a mainstream outcome used in several myopathies and even in respiratory diseases to assess performance of resistance in walking in patients. There were also secondary outcomes such as myotonia, strength, and some biomarkers. The results were very encouraging because they reported a significant increase in the primary outcome, which is the most important thing, an increase of distance performance at 6-minute walking test in the per protocol patients, 23 out of 40 with metformin versus placebo. Apparently, there were no effects on secondary outcomes, but it should be considered that the sample of this experimentation is quite small. They reported among adverse effects a higher frequency of mild to moderate gastrointestinal symptoms with metformin as opposed to placebo at this dosage. It should be pointed out that this is the maximum therapeutic dosage which is indicated for this drug, and this dosage probably causes a few dropouts in the trial. So this is very encouraging results as prompted us to expand this possibly this good result from using metformin. The great advantage is to use a drug which is already in use for decades, so safety concerns have been addressed for many years, and we know that severe adverse effects are rare, and mild adverse effects, especially on the gastrointestinal tract, may be quite common. So in 2019, we succeeded in having this study funded by a national drug agency of Italy, and the name of the study is the Efficacy of Metformin on Motility and Strength in Myotonic Dystrophy Type 1. Next, please. This is called briefly the METMID study, metformin in myotonic dystrophy. This is a phase 3B study is a triple blind since there is also blinding between the clinical rater and the physician taking care of distributing the drug and taking care of recording patients' impressions about the drug. This is due to the fact that metformin has a quite strong odor, so it could be detected by the patient, as opposed to the placebo, which is odorless. So triple blind, placebo controlled, on about 180 DM1 patients with a molecular diagnosis. Patients should be classified as MIRS-3 or MIRS-4 as clinical classification, and they are screened and recruited by 4 Italian neuromuscular centers. This is a non-profit study. As I said, it is funded by the Italian Drug Agency, and we administer 500 milligram metformin 3 times a day for 104 weeks. So as compared to the previous study by Guillaume Basset and colleagues, we are using a lower dosages for longer times. Our primary outcome is the same as in the French study, so the change in distance at 6-minute walking test. Of course, we point at a 40-meter change, a 40-meter gain in distance as a clinically meaningful measure for this outcome. We also look at secondary outcomes, which are quantitative muscle testing in the four limbs, CIS as a strength questionnaire, dexterity as assessed by the Knoll peg test, fatigue as assessed by the fatigue questionnaire, quality of life, and some biomarkers, among them oxidative stress biomarker, splicing analysis for a number of splicing products, and the detection of a number of RNA. Next one, which I guess, okay. This is just to show you the study flow chart, which shows here that after screening visits, we perform 5 further visits, which is 1 every 6 months, to the final visit, which is V5. And okay, next one, which I believe is the last one. The MedMe study is ongoing, so I'm just going to show you a few preliminary data, which is only demographic data, of course. So this data on screening and recruitment are as of August 31, 2021. The screening started at Site 1 in January 2020. Unfortunately, we had an interruption of screening due to the pandemic lockdown, which occurred between March and June 2020. Afterwards, we resumed the screening and recruitment of patients. And so far, we have screened 144. 141 were recruited. And among them, 93 are MIRS-3 as clinical stage, and 48 are MIRS-4. 81 are male, 60 female. Ethnicity is almost exclusively Caucasian, and the mean age for MIRS-3 is 43, and for MIRS-4 is 50 years old. And the mean 6-minute walking test performance at baseline was 371 meters for MIRS-3 and 255 meters for MIRS-4. The treatment is balanced among the sample. So far we have 70 patients on metformin and 71 on placebo. There were so far a few drug-related adverse events. None were severe, and in several cases we had moderate and sometimes mild and sometimes moderate adverse events, which were drug-related to our opinion, and most of them were pertinent to the gastrointestinal system, with mainly with diarrhea, some dyspepsia, and in few cases some weight loss, which was not very important. This is what I can tell you about this study, which is ongoing. Of course, we are going to complete the screening and recruitment of patients, which is going to stop probably at the end of this year, and so we have a long way to go still, because we're going to end the study at the end of 2023, with the last patients recruited stopping treatment, and the unblinding performed, and so hopefully next year I'll be able to give you more results, but we're not going to unblind the sample until the end of experimentation, and we hope that the initial very promising results obtained by Basset and colleagues will be confirmed and will be extended in number and in meaning, and so this next please. This trial is in the current pipeline of the experimental treatments for the myotonic dystrophy, standing as you can see here in a table produced by the Newcastle group, is right on the top, because being the drug a commercial drug, if successful, this and other similar trials with metformin will allow a clinical use of the drug. So this is the in the pipeline is the closest approach to the bedside for the currently experimented treatment procedures for the myotonic dystrophy. And I stop here, and I thank you all for the attention. And I am going to listen very carefully to the next speeches, which will bring us much more information. Thank you.
Thank you so much. Do you have any questions?
Well, I have one question. Roberto, hello. First of all, I enjoyed for your talk. I have only one question. In this ongoing trial, which I hope that will be very promising for our patients, you did not consider myotonia. So you look at mainly to the weakness.
There is a microphone which is open.
Can you give me an answer about this point? Thank you.
Yes, thank you, Gianni, for your answer. There is some audio disturbance, so no, now it's okay. I think there was some microphone going crazy, but thank you for your answer. Yes, we are not measuring myotonia. Well, this is of course it's a choice. Of course, it's a choice. I mean, since well, first we wanted to follow up with the encouraging results of Basset and colleagues, which show some good response on the walking resistance. So we intended to use the 6-minute walking test as a primary outcome. Secondly, myotonia is a bothering symptom for DM1 and DM2 patients, but there are several issues that we have to consider. First of all, is quite difficult to measure. There is a very high variability from day to day, and even within the same day, and among different between different seasons, and so it's quite different to compare. It's quite difficult to compare measurement of myotonia in time, especially in a long term. So we choose not to address the myotonia issue in this experiment. Of course, myotonia is important for myotonic patients, for myotonic dystrophy patients, and we have at least a symptomatic drug, which is mexiletine, which is going to be subject of a trial on DM1 patients, I think very soon, and which is used already as a off-label drug for myotonic dystrophy. But at least in Italy, we have the opportunity of having the drug produced by the Army chemical factory, and so we have the chances of seeing the good effects of mexiletine on myotonia. So yes, probably myotonia should be considered in future trials, but we wanted to focus on the resistance and on the muscle strength mainly.
Okay. Thank you. Thank you, Roberto. I want just to make another comment. According to mexiletine,
your microphone, Gianni, is off.
Yeah, okay. I want to just to tell you that now we have one paper from the French, from Sabine Vicard, coming out on neuromuscular disorders about the efficacy of mexiletine in DM1. Okay, just to confirm what you said about myotonic dystrophy type 1.
Yes, thank you for the advice, and of course this this is very important because we have to address this problem of myotonia, and either with a symptomatic drug or with a more general approach, which is targeting more upstream mechanisms that might influence myotonia as well, such as the expression of the chloride channel in chloride channel and the rear one, probably within the muscle fibers. Yeah.
Okay, maybe some comment from Guillaume.
Very briefly, and just to congrats Roberto and colleagues, since we do see here the question of the drug we're proposing. So in between symptomatic drug and highly specific molecular therapy that we heard, there is in this gap the metformin case, as it is for other repurposed drug. And then come the question, which is quite frustrating here because we're talking to patients, family, and an organization that the way is not more simple as compared to more complex therapy. And this is what we were facing in our country. And here I'm very glad that a national drug agency like the Italian drug came up and endorsed. You benefit from a very strong support that we did not at the time. So it's good that in this international network that there can be a place for a phase 3 clinical trial for one promising drug. Again, good luck, and we know how much effort, that this is a long road. 4 years for your case, we spent 3 years, so altogether it's nearly a decade to study metformin in DyMA.
Yes, thank you very much, Guillaume, for your encouragement, which we need so much. As you said, it's a hard work. It's a long term. We have to look for. So, as you know, accidents are always hiding in the dark, and they come out all at once with bad news. So we hope we'll have very few of these bad news. And yes, we were quite fortunate having this support from the National Drug Agency, although the budget, I can tell you, is not as large as one might think. And anyway, so we had to adapt our work to the budget which was made available, and so far we are succeeding in going on, and I hope I will terminate the study at the end of the experimentation with complete recruitment of patients and a good performance. So thank you for your encouragement.
Hello, sorry, I have one question. First of all, congratulations for all the the presentation, and of course for all the efforts during the last years, because it's amazing to have already clinical data and so much work done. I was curious about the biomarkers. Just I'm not sure if you can disclose any information, because of course you all know that looking for biomarkers that allow us to have some information about efficacy in the first stages and allow shorter clinical trials would be desirable and will be very helpful. So I would like if you can give us some information about the biomarkers that you were, the previous the ones that have worked better in the previous study, or if you are also planning to assess some biomarkers in the new study.
Well, as you may understand, I can't tell you so much, but some of the biomarkers we are going to look at are markers of the oxidative stress pathway, and we very much look with interest at the results by the Spanish group, by Muñain's group, which point to a exhaustion of the mitochondrial machinery. So this is one of our points. We want to look at the effects of metformin on the downstream markers of oxidative stress, and then we are going to look at some of the there is nothing particularly new here. Some of the known misspliced products of DM1 and mainly muscular misspliced products, and some of the microRNA which have been described by several groups in Europe, which are altered in muscular dystrophy type 1. So downstream effects of oxidative stress, well-known misplaced products, and most important microRNA in muscle.
Okay. Thank you.
You're welcome.
Okay, so just one question. You mentioned that the trial will take 104 weeks per people. How do you expect to maintain the commitment of patients for so long time? Patients who will support maybe secondary effects about the gastrointestinal.
Yes, thank you, Alain, for this question, which is really important, because we have experienced before, and we are experiencing the difficulty of having patients focused on an engagement which is so long term, such as 2 years. I mean, it's a risk. I agree with you. It's quite problematic to have focused patients which sometimes don't see overt amelioration of their symptoms. Of course, all those with most of those who take the placebo, of course, but probably also some of those who take the drug. So it's not a secondary problem. Okay, we are trying to face it with a very close follow-up with patients and families. Families are very important here, and so compliance is not always as good as we expect, but we are following up patients with periodic phone calls, asking for any problems which arise in between visits, and encouraging patients, and of course trying to cope with their main problems. The gastrointestinal disturbances, I believe, might be more frequent in this population of patients than in the general population, and this is because because the DM1 patients often complain of gastrointestinal disturbances, and so probably, unfortunately, this drug, this molecule, is targeting with a negative effect, of course, this kind of disturbances. We are, in most cases, we are successful in trying to correct these disturbances with symptomatic drugs, with regulators of the gastrointestinal motility, and in most cases it works. Of course, we had a few cases in which the problem could not be addressed properly. So I don't have so far data about dropouts, but I think we might have a few dropouts by the end of the trial, probably due to this problem. I hope this will be just a few, and so probably next time we can exchange and we can share some experience. I'll be able to give some more news about this.
Okay, thank you. We have to move on. As I said, we have plenty of time since the video from PepGen is only 10 minutes, so we are a little bit late, but that's not important. And move on to AMO. Thank you, Professor Massa. Mike Snap.
Hello, everybody, and thank you to Euro-DyMA for the opportunity to give you an update on our program today. I'm looking forward to speaking to you and taking any questions. Could you move the slide to the next one? Oops, a little bit of feedback. Very, very briefly, AMO Pharma is a small company. We are 9 people spread between the UK and the US. Our background is we are all individuals from the pharmaceutical industry, and most of the founders and management of the company have offspring who have what we call a developmental disorder. So some condition that affects the development of children meeting their milestones as they go forward. Sorry, could you go back to the previous slide?
Go back?
Go back if you, yeah. So our main focus is on developing novel therapeutic agents for the treatment of childhood onset diseases, mainly ones that affect cognition, that may produce symptoms reminiscent of autism, that are genetically determined and very rare. And our focus tends to be on molecules that will somehow alter the underlying biology of the disorder. We are less committed to purely symptomatic treatments. And the team, not myself, but my colleagues, have a great deal of experience in getting drugs approved for marketing with the FDA and the EMA. So a long history in drug development. Okay, we could move to the next slide, please. Our focus, by virtue of what I've just said, is on congenital myotonic dystrophy. That's not because we think that adult onset myotonic dystrophy is not important. Of course it is. But our skill set is mainly around technologies and methodologies to look at CNS symptoms. We're not really muscle experts, and we're trying to make use of the techniques that we've used in the past to get drug approved for pediatric neurological conditions. So hence our focus on individuals with the congenital or childhood onset form. Okay, we can move to the next slide, please. So AMO-02 is a traditional low molecular weight pharmaceutical. It's taken once a day orally as a strawberry flavored liquid. Once it's administered orally, it penetrates all tissues, so it accesses brain, muscle, GI tract, heart, and so on and so forth. It works in 2 ways. It directly reduces the amount of CUG RNA repeats within tissues, and it reduces the amount of a kinase called GSK3 beta. GSK3 beta is a molecule that works in opposition to muscle blind. Between the two, these, amongst other proteins, control the final differentiation of tissues, and the status of a tissue at any given time is a reflection of the balance between them. And the CUG repeat RNA in myotonic dystrophy, DM1, complexes with GSK3 beta and with muscle blind and disrupts the function of the two. What we know so far is that AMO-02 will reduce the amount of CUG repeat RNA and will reduce the excess levels of GSK3 beta protein seen in tissues as a result of the complex formed between the RNA repeat and the protein. We are presently investigating the mechanism by which this happens. We're writing up some data for publication and also looking at the effects on muscle blind because that complexes directly with the CUG repeat RNA anyway. In our initial preclinical work, we investigated cells from young individuals, infants in the 2 to 3 year old age range with congenital onset DM1, the HSR-LA mouse and the DMS XL mouse. And what you can see is some abstracted data from a publication. This is published as Wang et al. 2019. You can see in the pictures in the middle of the screen, the red staining is CUG RNA repeats, either in patient tissue or in the HSA mouse, and with administration of the drug that is removed. And to the right of that, you can see staining for the GSK3 beta protein, which again, the excess accumulated levels seen in patients or the mouse model are reduced by administration of the drug. And you can see in the panels below that, with administration of AMO02, we reduce the amounts of the CUG repeat and increase the amounts of breakdown products of that. And administering in vivo to mice, we can see a reduction in foci within muscle tissue with the drug. I don't have time to present today, but when we look at preclinical efficacy in both the HSR and DMSXL mice, we see benefits in vivo consistent with preclinical efficacy. And the most important thing I want to say with that is it's of relatively rapid onset. Okay, we could go to the next slide. AMO02, otherwise known as Tylglucid, was invented in Spain by a Spanish company called Nocera. They were interested in working in Alzheimer's disease and other neurodegenerative conditions, and they did a lot of work before they decided to move on from the molecule, and we took over development from them. The importance of their work is that we have an interesting amount of safety information on the molecule. So they administered it double-blind for periods of up to a year. We have since supported pediatric research in younger subjects with autism and have done a small phase 2 study in congenital or childhood onset myotonic dystrophy. And the main thing to say is that this is a pretty safe drug. It's generally well tolerated. The main adverse events of interest relate to asymptomatic elevations of liver function tests. And I'll show you some data on that later on in the presentation. But generally, an acceptable safety profile. Okay, we can go to the next slide. And we can go to the next slide. So I'm going to describe here a study that was run in Newcastle in the UK, 2017-2018. This was an initial study done under the supervision of the UK regulator, the MHRA. This was in individuals with congenital or childhood onset DM1. Their age range was 14 to 34 with a mean age of 22 years. It's a small study with 16 patients. And the way they were treated, they were put on placebo in the first instance to establish baseline measures. They were then transitioned to a high dose or a low dose of the drug for 12 weeks. Then they were taken off drug and followed up after 2 weeks to see what had happened once drug administration was stopped. The primary endpoint was safety, but we looked at a whole range of secondary outcomes. I'll talk in more detail in a moment. What we are mainly using as an outcome measure is a clinician-administered rating instrument that canvasses the whole of the phenotype of congenital DM1. We have a couple of caregiver measures that allow the caregivers to track on a very individual basis the symptoms that concern them most about the individual that they care for. We had clinician-administered ratings of general improvement and of autism, and then functional outcomes, the 10-meter walk test, and myometry to look at myotonia in hand grip. We haven't completed yet, but have been working on biomarkers and also looked at pharmacokinetics, the amount of drug that the individuals had in their system. And these data are now published as Horrigan et al. 2020, if you want to find the paper on them. This is the overall data. I'm not showing here what happened during placebo administration because there were no placebo responses, so there's nothing to see. If we look at the bottom of this slide, at the circles, these are pie charts showing the proportion of individuals who were responders according to the clinical global impression of improvement. As you can see, at the low dose, 6 out of 8 individuals were responders. At the high dose, 7 out of 8 were responders. And we believe we can explain why some individuals responded and some didn't, according to pharmacokinetics. So the individuals who were not responders had the lowest level of drug in their bloodstream. If we look up at the left-hand side, in the middle, the line chart here, CDM1-RS, this is a rating scale administered by the clinician, a pilot version of that rating scale. This is derived from the CCMD high, which is a measure, you can read publications on that, developed by Dr. Nick Johnson, now at Virginia. And it canvasses all of the phenotypes of the disorder. I'll show you some more data on that in a moment. What we can see is a clinically meaningful change compared to the end of placebo. This evolves quite quickly. We can see responses in a matter of weeks. It does not plateau over 3 months of treatment. So we imagine that if we treat for longer, we might see even more response. And it's dose dependent. In the middle of the screen, the line chart shows the top 3 concerns assessed by the caregivers. They can pick any symptom that they want to track, 3 symptoms. In general, they pick symptoms that are symptoms of congenital myotonic dystrophy, although sometimes they pick other symptoms like incontinence or something like that. And you can see again that over a few weeks of treatment in a blinded assessment, the caregivers show an improvement. And then the histograms on the right-hand side show mean performance at the end of placebo and at the end of drug for myotonia and for ambulation. Where there is a baseline deficit, the drug improves performance, but we should say most of the individuals in this study, their walking performance was quite good, and many of them did not have hand grip myotonia. So it's difficult to see what the drug is doing when there is not a baseline deficit. But overall, we showed on all of the key variables improvements due to the drug. So we were very excited by this, understanding that this is a small study with a specific design. Please, could we move to the next slide? So this is the initial version of the clinician-administered rating scale. We worked with the investigators and with the FDA to modify this scale and to make sure it is suitable and agreeable for use in phase 3 studies. You can see here the items that we focus on. They range from, I mean, you can read it yourself, but GI, muscle function, cognition, sleepiness, and so on. And the important thing to note here, our hypothesis is that AMO02, tideglosib, when taken orally, penetrates all tissues and addresses the biological basis of the condition by removing the CUG RNA repeats and fixing some of the complex proteins. If that's true, that should be happening in all organs of the body. So we should see symptom improvement in all organs. And this is what this breakdown shows. We see pretty much every organ system, symptoms related to pretty much every organ system improve with treatment, and most often in a dose-dependent fashion. We can move to the next slide, please. This is a set of plots. These are the individuals in the study. Each line is one person. These are the responders in the study. And this plots the amount of time it takes for them to respond. with the dark blue lines showing the highest dose. And you can see that the response is pretty rapid. There are clinically meaningful changes on the CGI scale within 2 to 4 weeks on the highest dose. So a relatively rapid reversal of their symptoms. Okay, we can move to the next slide, please. These are comments that were, during the conduct of the study, there's a text field that enables the clinician to capture comments made by the caregivers or potentially the patients to understand, what kind of changes are happening. And I've just picked some here just for illustration. Again, you can see these are young adults. So even though they've got congenital onset and have had their condition for a couple of decades, they're still capable of responding. You can see the comments that were being made by caregivers are very similar to the results obtained on the formal rating scales. So changes in cognition, more energy, less sleepiness, improvements in muscle function. And generally, what comes across when we review the whole of these data is improvements in what we would call activities of daily living. So individuals, being able to dress or express affection if they have autism symptoms, or do better in terms of cleaning their teeth or loading the dishwasher, that kind of thing. And this was very striking for us. And so we think it's important in our currently ongoing work to assess activities of daily living in the real world as well. Okay, please can you move to the next slide? So some of these data are published in poster form at conferences. We've worked with the University College of London to look at estimates of the total exposure to drug as assessed by pharmacokinetic measurements, plasma levels of the drug, and to see whether there are correlations with the clinical response to the drug. And although this is a very small study with a limited number of subjects, for all of the key outcome measures, there is a correlation between exposure over the course of the study and the degree of clinical response, which gives some more belief to us that the drug is what's causing the changes that are being seen in the individuals. Okay, we can move to the next slide. In terms of safety and tolerability, there's not a massive amount to say. There's a published literature from ourselves, Horrigan et al. 2020, and also Nocera published safety analyses in their longer-term studies in Alzheimer's patients. Generally, the drug is safe and well tolerated. The main adverse event of interest is increases in liver function tests. And if you could move to the next slide, please. You can see what happens with regard to those tests. So we see low-level changes in ALT and AST. These are never more than 2.5 times the upper limit of normal. There are no associated changes in bilirubin or any functional tests of liver function, no Hy's law cases, no individuals discontinued. What you can see, which is obviously a previously reported finding, about 60% of individuals with congenital DM1 have at least one abnormal liver function test level as a function of myotonic dystrophy. We see one individual in each group showing an elevation of greater than 1.5 times the upper limit of normal above their baseline. And also not shown here, some individuals who had abnormal levels, their, for example, CGT levels improved on drug treatment, maybe because we were reversing DM1-related changes in the liver. No individuals were discontinued as a consequence of this. You can see some of the individuals who have changes, their changes tend to normalize over time even on the drug, it settles down, and at the end of the study, all individuals go back to normal. Okay, we can go to the next slide, please. Please, can we go to the next slide? Although our focus is principally on congenital myotonic dystrophy, we have treated one individual with adult onset who has responded quite well in regard to muscle symptoms. So potentially this is generalizable as an effect. Can we go to the next slide, please? On the basis of the results we saw in phase 2, we're now running a potentially pivotal phase 2/phase 3 study. This is focused on individuals with congenital onset between the ages of 6 and 16. They will be treated in a double-blind, placebo-controlled fashion with a 1-to-1 ratio, half on drug, half on placebo. They will receive treatment for 5 months, and then at the end of that 5 months, they have the option, if they would like to take it, to transition to an extension study, which is an open-label safety study with 12 months of active treatment. The dose in both studies is the 1,000 milligram high dose, but adjusted for weight in children. We are using a range of outcome measures, measures of cognition, measures of muscle function, and then rating scale instruments similar to the ones that we used in the original study, the phase 2 study. So the primary outcome measure seeks to assay, as we mentioned, 11 items related to all organ systems scored on a 0 to 4 scale. And then I wanted to mention, we're looking at something called the Vineland Adaptive Behavior Scale as an exploratory outcome measure. That canvasses activities of daily living and how well the individuals perform in the real world. So we're interested to follow up on that because that was a striking finding in our phase 2 study. We can go to the next slide, please. The study, we were interrupted obviously by the pandemic. We had planned to start in 2019, but we couldn't get going until kind of April time this year. We are currently recruiting in the US, Canada, New Zealand. We're talking to a site in Australia and a site to the UK. Depending upon local conditions with the pandemic, that's where we want to open up. We are approximately halfway through the enrollment phase of the study. So the sites that are currently active and the families involved have done an amazing job of being able to work through the pandemic to keep going in the study. Our target, it's a target we hope we can meet, is to complete recruitment in the first part of next year. So we would have a readout something about a year from now. And then I mentioned the extension study will be ongoing after that. And I think that's my last slide.
Yes, it is.
Okay. So I'm happy
Thank
to
you
take
so
any
much.
questions.
Any question?
Maybe I can ask a question regarding how to link the quite large CDM rating scale to more tissue-specific objective outcomes. So what is your expectation on the patient feedback that they feel improvements of some functions? Is it more related to the CNS or the skeletal muscle tissue?
Sorry, the line's not terribly clear. The CDM1-RS looks at everything, muscle function, cognition, and so on. And then separately, we have specific measures in the phase 2 and in the phase 3 study to look at those separate domains, if you like. There is generally a good relationship between the two, but I would say the rating scale is more sensitive. So, for example, imagine an individual where we assess ambulation. Perhaps these individuals can walk 10 meters quite well. The question is, what happens if they try to walk, a third of a kilometer to their local school or shop or something like that, and then they begin to find it difficult? The rating scale tends to be a slightly better way of capturing that extra information. Also, for example, individuals who do not have measurable myotonia in their hands using myometry may complain of myotonia when they try to put their shirt on or something like that. And the rating scale does a better job, I believe, in capturing things in that way. And we've seen this in other similar rare genetic disorders before. So does that answer your question, or were there two questions?
No, I think the first answer, yes, was my question, to starting from a quite general and of course sensitive but non-specific rating scale to try to capture, to identify a kind of more target tissue-related effects, since the biodistribution of the compound is quite large. So it would be good to know, as part of the CNS or skeletal muscle or other, what would be the effect where of the drug, to try to maybe to have expectation in the future and to have better evidence of the effect. Since probably you didn't mention, or I didn't catch it, where was the GSK3 impact more observed? Is it in CNS cells? When you see patient cells, it's quite non-specific, so would be good to mention which type of cells. Is it muscle cells or neurons or whatever?
Again, I'm not sure I can hear terribly well, but the effects that we see, as you mentioned, the CUG repeat and muscle bind and GSK3 beta are probably disrupted in all tissues and all organ systems of the body. We believe the drug is accessing all of those. And when we look at patient performance, understanding that we've only done an initial study, we see improvements in CNS, improvements in muscle function, improvements in GI tract, and so on, either as items on the primary outcome rating scale, or when we use measures specifically analyzing function of CNS or muscle function.
Thanks.
Okay, thank you, Mike. Any other question?
Alan, can I ask a question?
Yeah, sure.
Yeah, Mike. So it was relating to the effects you saw on liver function, and also you said there was kind of a slightly dose-independent kind of response as well. So I was just wondering whether there was any correlation between those individuals that were metabolizing the drug faster and then the liver, the response from in terms of the liver enzyme activities.
I don't think we analysed that, Darren. You're right to say it doesn't seem terribly dose related. There was, if we set a criteria for being at least 1.5 times the upper limit of normal above the baseline, it was 1 in each group. It probably would be interesting to see if it relates to their PK levels. So that's a good point. We should take a look at it.
And a follow-up question would be, whether that's likely to be kind of genetically mediated by the known kind of drug metabolism genes such as CYP450, etc.
I don't think it is because the drug is metabolized polysymically by a number of different ones. So it's probably more likely a reflection of something a bit more idiosyncratic to do with the way it's absorbed in any given individual, or is subject to something like enterohepatic recirculation.
Okay, thank you.
Okay, if there is no more question, we will move to the next paper. It's from PepGen, and it's called Empowering Oligonucleotide Therapeutics. Unfortunately, the people from PepGen cannot be here today. They have delivered us with a video, and I will try to run it. You will probably see the images. I am not sure you will hear the sound. Let me know.
Hi, my name is Jane Larkindale, and I'm the Vice President of Clinical Research at PepGen. I really wish I could join you at the Euro-DyMa meeting this year, but unfortunately, at the last moment, we were unable to be there in person. So I really would like to thank the organizers for giving us the opportunity to record this talk in advance of the meeting, so at least we can give you some idea of what we're doing at PepGen and how we're developing a really exciting new therapy for myotonic dystrophy type 1. So a huge thank you to the audience. And if you have any questions, please contact me, and I'm happy to talk offline at some point. PepGen is a company that's really focused on delivering oligonucleotide therapies for neurological diseases, and in particular neuromuscular diseases. I think many of you will know about oligonucleotide therapies. These are small pieces of nucleic acid that interact with the genome inside a patient and really change how diseases progress. We're particularly interested in neuromuscular diseases and neurological diseases, which have been greatly challenged through the years by delivery problems. The oligonucleotides work well in cells, but not enough of them ever get to skeletal muscle, to cardiac muscle, and to smooth muscle, which are the areas where we really need to get to have an effect in diseases like myotonic dystrophy. So that is what our company's platform is based on, is technology that helps us get those drugs to where they need to be. And I'm going to show you some data that shows that we believe we've really managed to do this. So this is our pipeline. We're an early stage company still in preclinical development at the moment. Our lead program is in Duchenne muscular dystrophy, and our second program is in myotonic dystrophy. But that does not mean that we are ignoring myotonic dystrophy. We are moving that program forward as fast as we can, and we hope it will be in the clinic by 2023. So how are we doing this? This is really the work of, for many, many years, from our academic founders at the University of Oxford and the University of Cambridge. We know that oligonucleotides can have a profound effect in cells. That is a well-established fact, but they haven't been able to get there. And we have been developing these enhanced delivery peptides, these little bits of protein that we attach to the oligonucleotide, and which makes it really go into cells, particularly in muscle cells, cardiac muscle, skeletal muscle, and smooth muscle. So we call these enhanced delivery oligonucleotides, and we're really excited to be moving them forwards towards the clinic. This is some data that comes from our early academic founders. What you can see on the left is a single injection into one of the leg muscles. If you inject saline, nothing much happens. This is actually a model of Duchenne muscular dystrophy, but this was true for whatever the target of the oligo is. The initial set of peptides that were developed, these PIP-PMO conjugates, really showed much better skipping in muscle. It was getting into muscle way better than oligos that didn't have the peptide on, which was great, except for one thing, which is if you look over on the right-hand side over here, you can see this thing called KIM1, which is a marker of kidney damage, was also going sky high. So these peptides were really effective, but they were also toxic, which doesn't make for a good drug. So the founders of our company spent a lot of time playing around with chemistry, and that's what you'll see in the blue bar, where you can see that the DPEP is nearly as effective at getting into muscle as the PIP, but that toxicity is gone, and we can go back, it looks equivalent to saline, and we have a drug, something that could actually be used as a drug, and that is the basis of the work that we've been doing at PepGen. This is data just showing what how much of the PMO, that's that oligonucleotide, gets into different parts of a monkey. You can see on the left, skeletal muscle, various different skeletal muscles, all of which are important to myotonic dystrophy. We get into the diaphragm. Very importantly, myotonic dystrophy, we get into the heart, which has been a real challenge for oligonucleotides through the years. We get into smooth muscle extremely well, that's your gut, where again, many myotonic dystrophy patients have a lot of symptoms. And we even get into the central nervous system, into the brain to some extent. Again, a really important target in myotonic dystrophy. So we think for a disease like myotonic, which is so multisystemic, where so many of you living with myotonic have issues or complaints in all sorts of body systems, a technology like this that gets to all kinds of different parts of the body is going to be really, really effective. So with that, I'm going to switch over from our platform to talking specifically about the molecule we call PepGen Euro-DyMA, which is targeted for myotonic dystrophy type 1. So I think that this is a rather complicated slide. It actually tells you a lot of things that you probably already know. In myotonic dystrophy type 1 patients, there is a CUG repeat, just a repeating sequence inside the sequence of a protein called DMPK. That sequence forms what's called a hairpin loop, and that binds to a protein called muscleblind. It doesn't matter what they are, but the downstream of this is that when the muscleblind is bound to the repeat, you get a whole lot of downstream issues that causes all the disease problems. Our Euro-DyMA binds to that repeat. So it sits there instead of the muscleblind, leaving the muscleblind to go off and do exactly what it normally does in everybody else. And at the same time, we don't believe it leads to DMPK degradation. So the gene where the repeat is fine, the muscleblind is released, and we think this should have a profound effect on myotonic dystrophy symptoms. So I'm going to just show you the results from a couple of experiments that we've done in a mouse model. This isn't exactly myotonic dystrophy type 1, but it has a repeat sequence. It's in a different gene. And very importantly, it shows the same symptoms as people with myotonic dystrophy. You sequester the muscleblind, you have downstream of splicing events, and you have myotonia that we can measure. And you can see the myotonia over here in the pictures, that a resting mouse looks just fine, but after it's been pinched, you can see these legs sticking out behind. They are myotonic, they're stiff, and it takes a little bit before they can start functioning again. So we looked at this model, and you can see over here on the left, some of the splicing changes. These are just how individual pieces of gene are processed. You can see in black wild type, this first graph shows ATP2A1, which is just one transcript. You can see without our drug, this exon is not included. And with increasing amounts of our drug, you start going back to normal levels of this exon being included. This is a different one, CN1. It's the opposite thing, that normally in wild type unaffected mice, this is there's 0% inclusion. In an HSA mouse, you have quite a lot of inclusion, and with increasing amount of drugs, it goes back to pretty near normal. So this is the splicing effect that we see in myotonic dystrophy in humans and in the mouse. And you can see that our drug, as the concentrations go up, basically corrects that completely. On the right-hand side over here, you can see myotonia, that wild type mice take about 30 seconds, around about 30. With no, the HSA mouse, you can see it's much slower to relax. And then with increasing amounts of drug, we get to complete correction of that myotonia. And I'm going to show you that in the next slide, because a picture or a video in this case is worth 1,000 words. So on the left, here is our untreated mouse. This is the mouse model of myotonic dystrophy type 1. And if I play the video, you can see as it's released, it's pinched and its legs stick out behind it, and it takes a little bit before it can relax those legs and start to run on them. If you look on the right, here is a mouse after a single treatment with Euro-DyMA. And you can see it almost immediately starts running. There's no myotonia. The legs work just fine. So over here, you can see the legs dangling behind the mouse for a little bit. Here, absolutely no myotonia at all. And this gives us a lot of confidence that this treatment is going to work. We can treat the downstream effects of myotonic dystrophy, at least in this mouse model. How long will it last? This is a separate experiment where we looked at those splicing changes we were just looking at, not just over a short period of time, but over 6 months. And you can see that after a single injection, 6 months later, we still have correction of those splicing changes, which gives us confidence that we should be able to dose in humans not too frequently. We're not going to be asking people to come in for an infusion every week. And hopefully this will have a long-lasting effect and increasing effect over time on your symptoms. This is just going back to the figure I showed earlier, that what we've just shown you is work in the muscle, because that's what we look at in the mouse. But we know our oligo also gets into heart, into smooth muscle, and a little bit into the CNS. So we have hope that we will also be able to correct symptoms in those systems as well, and have a much more profound effect on the symptoms that really matter to people with myotonic dystrophy. So where are we? Well, we've just finished a whole lot of mouse studies. I've shown you some of the data from that. We are validating some of this work in human cell lines, which have different repeat lengths, because we all know not everybody with myotonic dystrophy has the same length of the repeat. And then we're going to initiate all the formal studies we have to do to go to a global regulatory agency and ask to start a clinical trial. We're really expecting to get through most of this work next year in 2022, and our goal is to get into the clinic and start our first clinical trial for myotonic dystrophy type 1 patients in 2023. At the same time, we're not resting on our laurels. We think we have a really good technology now, but you can always do better. Right now, we're focusing on our Duchenne and myotonic dystrophy type 1 program, but we're going to look into what other targets we can look at. And I know many of you may be interested in myotonic dystrophy type 2, and we may go there, we may not. We're still working on that. We're also designing new peptides so we can increase our ability to get into the brain, increase our ability to get into the heart, and really make sure we can have as robust an effect as possible with future programs. So the power of our platform is really our ability to deliver these oligonucleotides to all the sites in the body that matter. So far, we have data showing we have robust activity and really good tolerability. And we're moving forwards to get this into the clinic as fast as we possibly can. And with that, I'd just like to thank our team, and particularly the Euro-DyMA team, for letting us speak today, and particularly for letting us record this in advance so we could share these exciting updates with you. I'd love to hear from you. We'd love to talk to you. We've been reaching out to the myotonic dystrophy community to understand what's important to you, how we can run trials to make them as convenient as possible for you, and what symptoms are most important to you. And we'd love to hear from you. Would love to continue working with you as a community as we move forwards. And with that, thank you very much, and I hope you enjoy the rest of this meeting.
We have to move on to the last talk from Dine. And here it is.
Wonderful. Thank you, Alan.
Okay.
Can you hear me okay?
Yes, it's okay.
Okay.
At least for me.
I'm live, no video, so you have me. I'm actually speaking here.
You don't have any video? Wow.
Wonderful.
And the others?
Okay, I'll get started. First of all, thank you very much for inviting me, Alain, and the organizers to this meeting. This is fantastic. Very happy to be here. So just by way of introduction, I'm Ash Dugar, so I'm the Global Head of Medical Affairs at Dyne Therapeutics. By way of background, I've been at Dyne now for about 9 months, and so doing my best to learn as much as possible, as fast as possible in myotonic dystrophy. And so these kinds of meetings are incredibly valuable to me, so thank you for inviting me. Prior to that, I spent some years at Sarepta, focused on Duchenne and limb-girdle muscular dystrophy, Charcot-Marie-Tooth, and other disorders such as that. So I'm very pleased to be here today. For the next 20 or 30 minutes or so, I'll talk about our FORCE platform and data from our myotonic dystrophy type 1 program. And at any time, please stop me if you have questions, or I can take them at the end. If you can go to the next, oh, thank you. So this slide just shows our forward-looking statements. You've seen this slide, I'm sure, in many forums and for many companies, but I just want to take a moment and put this up here. Yeah. Next slide, please. A little bit about Dyne. So I think it's important to describe our mission. At Dyne, we are really focused on delivering life-transforming therapies to patients with serious muscle diseases, with myotonic dystrophy being a if you can go back to the previous slide. Thank you. With myotonic being a priority. And our mission is core to who we are and what we do every day. It's one of the reasons that I joined the company. I think it's truly focused on the science and on the patients. And we're trying to do the most we can to understand the community of people living with muscle diseases and DM1 in particular, and ensuring that the patient's perspectives and the physician's perspectives and experiences are included in our drug development program. And many of you may know Molly White, who was the head of the Myotonic Dystrophy Foundation for many years, and she also works with me very closely. She's at Dyne now. So we've really created a multifaceted program that's involving community webinars, conference presentations, fireside chats, and other outreach to raise visibility and gather input to advance our drug development efforts. So over the past few years, we've been validating our platform and technology, including the ability to target delivery of potential ASOs to muscle. And I think we've generated some really exciting preclinical data across multiple indications, including DM1. And our focus today is on sharing the data from the DM1 program. Next slide, please. Briefly, we are building Dyne as the leading muscle disease company to deliver for families living with DM1, and as well as other muscle diseases like DMD and FSHD. So on this slide, we show our 4 core strengths to help us deliver on that promise. We've intentionally designed our FORCE platform to overcome the current limitations of muscle tissue delivery with modern oligonucleotides. And the goal for our DM1 program is to develop a therapy that will lead to reduction in toxic nuclear DMPK, correct splicing deficits, and enable relatively infrequent dosing to potentially stop or reverse disease progression. Second, our focus on rare muscle diseases. We've generated very exciting preclinical data that shows the power of our technology across diseases and in DM1 in particular. And I'll highlight just some of this data in the presentation. The third, as I've mentioned, it's delivering for patients. We're going to leverage our muscle disease focus to advance our programs as fast as we can to the clinic. We've shared that we plan to submit 3 INDs between the end of this year and the end of next year, with the DM1 program anticipated at the front part of that timeline. And then we have great in-house and advisory expertise with deep knowledge of DM1 and novel therapeutics and ASOs, and a world-class scientific advisory board, which include luminaries such as Dr. Charles Thornton. Next slide, please. Thank you. So our FORCE platform consists of a specific delivery mechanism called a Fab, or an antigen binding fragment. And we've engineered this Fab to bind to a receptor called TFR1, transferrin receptor 1, that is found across the surface of muscle cells. So if you look at the left-hand side, the top left of this slide, at the Y-shaped structure on the left, that's of course a monoclonal antibody. Yet our FORCE platform uses only that top piece, uses the antigen binding fragment, or the Fab, which has a high affinity to the TFR1 receptor, and that drives binding to the TFR1 receptor on muscle cell walls. The TFR1 receptor is highly expressed across various tissue, including skeletal, cardiac, and smooth muscle. And we believe the Fabs have several advantages over full antibodies, or mAbs, including their smaller size, which helps with tissue penetration and distribution to key muscle tissue. So essentially, the Fab acts as a kind of key that unlocks that TFR1 receptor and allows our therapeutic oligonucleotide to enter skeletal, cardiac, and smooth muscle to target the genetic cause of the disease. Also on this slide, you can see that we link our Fab to the payload with a linker. Now the linker molecule connects the Fab to the antisense, which gets in the nucleus and is designed again to target the genetic basis of the diseases we're trying to treat. And this linker is clinically validated. It's been used in products that are actually on the market, so it's very well understood We understand how to enable precise conjugation, and the kinetics of the linker are also very well understood. And in the case of DM1, as I said, our potential therapy targets nuclear DMPK RNA, and I'll discuss that more in upcoming slides. As you know, historically, the challenge, as stated earlier from me and other companies, has been the inability to get enough of these ASOs into muscle cells. And our FORCE platform is designed to solve this specific challenge, to solve the delivery issue, to be able to deliver the ASO into the muscle. So moving on to slide 6, we believe the FORCE platform provides significant advantages over other approaches by delivering targeted, redosable, titratable, safe and effective therapies which can be used chronically. We've also demonstrated extended durability, even up to 12 weeks, and we're currently exploring longer effects to enable less frequent dosing. So we think it's going to be a compelling product profile for patients. The other nice thing is we use the same Fab and the same linker across all of our programs. So that does reduce development timelines as well as manufacturing costs. Next slide, please. So turning to slide 7 here, we have built a robust portfolio focused on muscle diseases. Again, DM1 and FSHD represent opportunities to develop first-in-class therapies, and in DMD, we're building a global franchise, starting with our exon 51 program, followed by the development of therapeutics for about 80% of patients with mutations amenable to skipping other exons, including 53, 45, and 44. So we're really excited about the utility of this platform across muscle diseases. Next slide, please. We're really excited about the progress of our DM1 program and its potential to deliver disease modification for patients such as Joachim, pictured here on slide 8. He's a friend of Dyne, diagnosed with DM1 in his 30s. He's now in his 50s and lost his father to DM1. Everyone here knows the disease much better than I do. Clearly, DM1 presents with myotonia and muscle weakness, as well as cardiac and pulmonary abnormalities. And there are many patients in the US and Europe and elsewhere that suffer from DM1. And right now, our goal is to stop or reverse the progression of the disease, and hopefully our platform is able to do that. Next slide, please. As I said, FORCE targets the genetic basis of disease. I won't go through the slide in detail. Essentially, as you've heard, as you know well, in DM1, the toxic number of repeats form those large hairpin loops that remain trapped in the nucleus, forming nuclear foci. And I bring this up because the data I will show you in a few minutes, I'll come back to this theme of being able to reduce nuclear foci. So the toxic nuclear RNA acts as a sponge which binds those splicing proteins, forming the foci and causing abnormal splicing events in many genes that are important for muscle health, such as chloride channels, calcium channels, and other muscle structural and functional proteins such as BIN1. And again, I'll come back to some of this because I will show some of the data related to these factors. And this abnormal splicing across multiple genes leads to the clinical phenotype that you know very well for DM1. Next slide, please. So as I get into the data, I'll show some in vitro data as well as in vivo data. Here, what we show is our lead candidate being studied in DM1 patient cell lines with approximately 380 repeats, and on the next slide, it'll be 2,600 repeats. In this patient cell line with about 380 repeats, our candidate demonstrated a robust DMPK RNA knockdown and reduction in nuclear foci, as represented by the reduction in the red foci on the right-hand panel shown here. We also demonstrated a correction of splicing defects as measured by BIN1 exon 11 inclusion, as shown in the histogram in the middle of the slide. So this just tells that it shows that entire picture of DMPK knockdown, splicing correction, and nuclear foci reduction. And on the next slide, please. Great. So here we have a DM cell line with about 2,600 repeats, which represents a severe disease phenotype. And here we also saw robust dose-dependent DMPK RNA knockdown, reduction in nuclear foci, as well as BIN1 splicing correction. So we're really pleased by this in vitro data that gives us a tremendous amount of confidence to move forward into our in vivo studies. Next slide, please. I think you've heard about this model, you know about this model very, very well, and some data from another sponsor was provided. Here, as you know, the HSALR model has CUG repeats in the nucleus, and so it mimics RNA missplicing that's observed in patients. And it is one of the very few models that presents with a myotonia phenotype. So it's quite a useful tool to be able to look at different compounds. As shown on the left panel here on slide 12, we observed dose-dependent correction of splicing in multiple muscles, including the quadriceps, gastroc, and TA. And thereby we reversed myotonia, which is shown on the right panel. So the red circle is a vehicle-treated cohort of mice. Green represents 10 milligrams per kilogram, and blue represents 20 milligrams per kilogram of force administered IV. And what we observed was a dose-dependent splice correction towards wild type with that single dose of the force compound. Next slide, please. We also developed an innovative HTFR1 DMS-XL mouse model to evaluate both pharmacodynamics and pharmacokinetics. We think that this model really sets a new standard for evaluating PD in myotonic dystrophy type 1. As you see on the left-hand side of the slide, this model represents a severe DM1 phenotype with more than 1,000 repeats. And you'll see that the mice in this model express the human TFR1 and carry a human DMPK gene. So if you move to the right-hand side of the slide, you can see how this enables us to evaluate the ability of our lead DM1 candidate to reduce toxic human nuclear DMPK RNA, since the model expresses the human TFR1 receptor. So that, quite a lot of utility with this model to be able to understand what's happening with toxic human nuclear DMPK. Next slide, please. So I'll take you through a little bit of data using this mouse model. There's many genetic studies in the field that have indicated, hypothesized, that aiming to target 30 to 50% knockdown in toxic DMPK has the potential to be disease modifying. So on slide 14 here, you'll see data that we've reported previously where we've evaluated our candidate in the HTFR1 DMS-XL mouse model after 2 doses, each dose at 10 milligrams per kilogram and evaluated at day 14. Our candidate demonstrated a significant toxic human nuclear DMPK knockdown in various tissue, 60% in the heart, 56% in the diaphragm, 54% in the TA, and 39% in the gastroc. So we believe this represents a very important finding and robust knockdown of the DMPK in this mouse model after 2 doses and measured 14 days later. If you turn to the next slide, so the histogram on the left-hand side is the heart knockdown data that you just saw on the previous slide. Additionally, and consistent with this, on the right-hand side of the slide, you can see that our candidate demonstrates an approximately 40% reduction in DMPK heart foci at 14 days. We believe this is, absolutely critical to be able to demonstrate that we are able to get into cardiac tissue and affect a change. And in this case, looking at the foci reduction in the heart. So, very important that we're targeting all key muscles, and I think this is quite important data that we're generating. Next slide, please. So we expanded our analysis in the HTFR1 DMS XL mouse model to evaluate treatment with a single low dose of 10 milligrams per kilogram and at a longer time period, as you can see on slide 16. In these data, our candidate demonstrated sustained reduction in DMPK at 4 weeks, 51% in the diaphragm, 46% reduction in both the heart and the TA, 42% in the gastroc, and 42% in the gastroc. And also in these studies, it's important to mention that our candidate was very well tolerated. So what this shows us is that we see very similar robust knockdown of toxic human nuclear DMPK RNA that is sustained with twice the duration and at half the dose that you saw 2 slides ago. So we think this reinforces the advantage of the FORCE platform to enable targeted delivery to muscle of oligonucleotides with relatively low and infrequent dosing. Next slide, please. So just to recap, again, we've designed this platform to address the genetic basis of myotonic dystrophy type 1 by targeting toxic nuclear DMPK RNA. And the data that I've shown demonstrate that a fully human FORCE conjugate can correct the DM1 phenotype of patient-derived myoblast cultures with a range of repeats, including those that represent severe DM1. We can reduce toxic nuclear human DMPK foci in cardiac muscle of the HTFR1 DMS XL mouse, and we show sustained knockdown of toxic nuclear human DMPK in this mouse model after just a single dose. And all of this work, along with additional non-clinical work, both in rodents as well as non-human primates, is strongly supporting further development for our DM1 therapeutic candidate, including a planned clinical study. If you can go to the next slide, please. And one more, please. Sorry, just wanted to make sure that I showed the acknowledgements. So thank you very much for your time, and I'm happy to take some questions.
Okay, thank you so much. Time for questions. Yeah. I'm sure you have plenty of.
I'll do my best to answer them. And if I don't know the answer, I will take it down and I will get back to you, Alain, with answers to those questions.
No question at all? So yeah, one, you mentioned the entry in clinical for a planned test in the next years. On your website, it's mentioned between end of 21 and end of 22. Do you maintain this information?
Absolutely, yeah. We are confident in the guidance, and so the other thing that I would add is the DM1 program is at the front end of that range, and again, everything is moving on track, and we're looking forward to getting our IND out there and starting clinical trials. So, but everything is moving according to plan right now.
Okay, good. Thank you.
And we're also, I will just mention, we're also doing a lot of work with the patient community. We have advisory boards. We meet with them regularly to get input into our clinical program thinking, as well as some of you that are on the line here and others have been very generous in telling us your thoughts on what to think about as we develop our clinical development strategy. So thank you.
Okay. Questions? Yeah, here.
Yeah, can I ask regarding encouraging knockdown with a long duration? Is there any thought on administration, a route of administration by subcutaneous rather than IV, or did you test?
Yeah.
Yes.
It's a great question, and we've been thinking quite a bit about that internally. So currently, we've done experiments only with IV administration in the various preclinical studies that we've done. We are thinking about subcutaneous because we think that would provide some significant decrease in patient burden, which would be fantastic. And so that work is ongoing in terms of just trying to understand the best way to start to develop a sub-Q formulation. So we've literally been having these discussions over the last 2 months or so, and so we are still sort of planning that plan for developing a sub-Q. So we'll update you when we're ready to update you. But for now, it's IV administration.
Any other question? Okay, there is no. Well, thank you again. It's the last topic that we wanted to address in this first meeting, first Farmers Day, and we are perfectly on time. So now it's time for going back to more generally, what is your thinking about this meeting? What are your suggestions for next year, same meeting? And it's open for discussion. No one? Nobody wants to speak? Okay, good. So if there is no comment, if there is no question, I thank you everybody for your participation and hope to see you in the next month, I am sure, for some of you. But I thank you. Bye.
Thank you.
Thank you so much for the organization. Goodbye.
Bye.
Bye.